I'd like to thank Christian for inviting me. It's an honor to be here. Our group does mainly 3D structural modeling and also wet lab biochemistry, and then we're just kind of tiptoeing into RNA bioinformatics. So it's wonderful to be here to interact with so many people in bioinformatics. So today I'm going to talk about structure-function relationships in individual long non-coding RNAs. And long non-coding RNAs are a new class of emerging RNAs that have been basically come into their own as a field in the past 5 to 8 years or so. They tend to be long in length on the order of kilobases and really critical for many processes important in development and epigenetics and in brain function as well. So first, the motivation here is basic epigenetics and RNA biology. So in epigenetics, there are many outstanding issues these days. If you're not familiar, epigenetics is when you have an inherited change in phenotype without an inherited change in genotype. And there are many, okay, so there are different mysteries such as how does the DNA get demethylated is one. The mechanism of stem cell reprogramming isn't really well understood in gory detail right now. Inheritance of histone modifications and also therapeutics are big outstanding questions. But one of the other interesting questions is the mechanism of recruiting epigenetic factors to specific sites on the chromatin. So we know that the chromatin has to be modified in a cell type specific way and also in many cases a developmental stage specific way. But how does the cell know how to do it at that place at that time? And some people are speculating that it could be RNA, could be the answer to this. So an RNA could be transcribed off the DNA, and then its fold could be dependent on the environment, such as hormones and different factors there. And then a specific fold could recruit one of those factors. And so what we're focusing on are these RNA molecules. And so what is a long non-coding RNA? A long non-coding RNA is typically classified to be anything longer than 200 nucleotides, and in practice in eukaryotes and more, what most people are interested in are in mammals and humans. And these are often, even though the definition is greater than 200 nucleotides, they're more typically between 1 to 10 kb in length. And some of the important ones are actually on the order of 100 kb, so they can be quite large in length. Yeah. These, well, it kind of depends on your definition. So, for example, there is another exciting class of RNAs called small RNAs in bacteria that are longer than 200. Many are longer than 200 nucleotides. So there can be some mix there. And these ones that people are studying in the field of long non-coding RNAs they often look like a regular coding RNA. They're polyadenylated, they're capped, and they're spliced. But people can't find a protein associated with them. And interestingly, there was like a kind of a controversy about 3 or 4 years ago where people are saying, well, maybe these non-coding RNAs actually code for tiny peptides. And then there was a lot of work doing ribosome profiling to look at the signature of ribosomes on the mRNA, and they found different signatures for coding versus non-coding RNAs, so the distribution of ribosomes along the RNA. So in particular, at the stop codon for a coding RNA, you would have like a sharp drop-off where all the ribosomes were gone after that. But in the case of non-coding RNAs, it was a more gradual shoulder-like feature. And then these RNAs can act in cis or in trans, and they're often associated with epigenetic marks. So one of the main marks is this H3K27ME3, the trimethylation of lysine 27 on histone 3, and many other different epigenetic marks are associated with these non-coding RNAs. Whoops. Okay, so while many of the groups looking at long non-coding RNAs are doing genome-wide studies, looking at across the genome, for example, how are long non-coding RNAs affected by estrogen, or looking at cancer versus non-cancer. Our tactic is to look at just one non-coding RNA at a time. And there's this Japanese proverb, a difficult task cannot be done quickly. It must be achieved gradually by small steps, a little at a time. And this is printed alongside one of these tempos where I think there are 500 steps, and then that's the lesson you're supposed to learn as you're walking up the steps. So we're looking at these RNAs one RNA at a time. And we're inspired a lot by what's happened in the ribosome field over the past 40 years or so, since the ribosome is sort of one of these original long non-coding RNA systems. And we've done a lot of work in structural modeling of the ribosome. In 2002, we did some modeling of the E. coli ribosome, comparing to one of Joachim Frank's cryo-EM maps. And then we also came up with one of the first structural models of the intact ribosome in 2004. And then more recently, we've been producing all-atom structural models consistent with cryo-EM data for different translocational intermediates, like in E. coli, for example. And then in addition, we also do a lot of all-atom molecular dynamics simulations of ribosomes. And so this is just kind of advertising some of the other work we do in the lab. This is a microsecond MD simulation, explicit solvent, and we use the HPC platforms in Los Alamos. And so here we're looking at the atmosphere of ions, potassium, chloride, and magnesium. And then if we disappear the ions, we can look at the ribosome itself. And this is looking at different scales of time steps. And so we can see the small and large subunits, and then the darker colors are the RNA, and then the lighter colors are the ribosomal proteins. This is the L7, L12 stock. And in the simulations, we get a nice stable MD runs of the ribosome, where, however, the parts you expect to be flexible, as shown in single molecule experiments, these are flexible in the simulations as well. But in the core, we get nice stable RNA helices and so forth. And these kinds of simulations allow us to look at fluctuations of the ribosomes, which were the sort of fluctuation hotspots, and to try to make contact with single molecule FRET data. And this was a big calculation. We did this about, we finished the runs over 5 years ago now. And so we got to a microsecond 5 years ago. So nowadays we can do much more than just a microsecond. And this will go out to the 1 microsecond, yeah. And then just another, whoops, I'm going fast here. Okay, let me flip back. So this is a fit to a cryo-EM map. We do a lot of fitting of all-atom structures to cryo-EM data. And this was also done back in 2004 for the human ribosome. And nowadays, the resolution, this was, I think, around 6 angstrom resolution. And nowadays, you can go to much higher resolution, so-called atomic resolution. And then this was just saying that the ribosome is sort of one of the original gangsters of long non-coding RNAs. So the ribosome, it's one of the only big RNAs that's been studied to the point where you can get an all-atom cryo-EM or X-ray structure and also do single molecule and molecular dynamics simulation for one of these giant RNA protein systems. And so we're using this as kind of a template for our work on long non-coding RNAs. And so the idea is that in the 80s, they came up with the sequence for the ribosome, and then the next step was to look at the secondary structure map of the RNA of the ribosome. And this was work done by Harry Noller, Carl Voice, and Robin Guttel and others back in the 80s. And then after the secondary structure is done, the next thing you can do is try to get 3D information, sort of very coarse grain 3D information through scattering, neutron scattering or small angle X-ray scattering. And then with all that data, you can go forward with cryo-EM and single molecule and the more detailed structures to try to get an idea of the mechanism on actually how that RNA works. And so in the long RNA field, we're kind of in this area now where some of the first secondary structures are being mapped. Our group produced one of the first secondary structures of a long non-coding RNA. And then people are just sort of tiptoeing into trying to see if there's 3D, are there tertiary contacts, is there 3D information associated with these molecules. And this is just showing the 16S ribosomal RNA. And the goal is to come up with these kinds of maps for the long non-coding RNAs. And these, I want to emphasize, are really critical when we're thinking about trying to solve 3D structures of these molecules. And the reason is that if you have a high-resolution cryo-EM map, it looks very noisy, and it looks like a mess, basically, if you have thousands and thousands of little blobs everywhere. And if you don't even know the secondary structure of the RNA, it's pretty much impossible to come up with the 3D structure of it. So the secondary structure is really critical to give you a framework for moving forward with the 3D structure. And so what are some of the issues in the field of long non-coding RNAs? So the first major issue is, do these RNAs have any structure at all? So right now, or when the field first started, it was almost entirely cell biologists, and I would say many of the cell biologists are skeptical. I think many of them believe that these RNAs are just almost single-stranded and just kind of entirely disordered. And then there's some more biochemists and a few structural biologists entering the field who believe they're completely structured and look like ribosomes. And so probably the truth lies somewhere in between. Now there are about over 10,000 of these RNAs that have been discovered. So we're betting that of the 10,000, at least 10 or 100 of them are going to have really nice 3D structures. So this is kind of our game plan. And then another big question is, are the RNAs conserved? And so by sequence alone, most of these RNAs have pretty low sequence identity. But as we heard in Christian's talk, there's more to conservation than sequence identity. You can have conserved structure with large variation in sequence. So the structure and function can still be conserved even if you have a non-conserved sequence. And then finally, the ultimate goal is to look for structure-function relationships, so try to understand how these molecules really work. And so again, do these RNAs have well-defined structures or not? So you can imagine a disordered kind of RNA or a highly ordered one, or maybe something in between. And then these are kind of just artists' renditions of a disordered one that might have a small pocket of protein binding sequence. You can imagine a highly ordered RNA with a well-defined binding site as well for proteins. And then you could imagine something in between with a more spread out RNA that can bind many different proteins or something like this. There are more possibilities. These are just some of the possibilities. And then there are many other groups working in this field. Most of them are focused on genome-wide studies of secondary structure in the long non-coding RNAs. And then there's a few groups, like our group, doing secondary structures of just single individual RNAs, like our group, Anupal's group, Chang's group, and a few other groups are now doing single RNAs. And so now to talk a little bit about this question of, are the RNAs conserved? Most of you may know that in terms of protein amino acid sequence identity, you may consider a conserved protein to have, say, 30% sequence identity. And then if you do blast searches, often the defaults are set around 95% sequence identity with maybe 40 contiguous nucleotides. And many RNAs satisfy these criteria that you can find in something like BLAST, like the microRNAs or the ribosomal RNAs. However, there are other RNAs where you would pretty much never find with a BLAST search that have low sequence identity, like the riboswitches, U2, U4 spliceosomal RNAs, the 5S ribosomal RNA, and the group 2 intron. And so if we look at the riboswitches, these are non-coding RNAs mostly in bacteria. And these RNAs bind small molecules, metabolites, and they can control gene expression by folding differently whether or not the metabolite is present. And so Ron Baker at Yale discovered almost all the riboswitches through a combination of chemical probing in the wet lab, and then he would basically solve the secondary structure with chemical probing, which I'm going to talk about in the next few slides, and then do some sequence comparisons to see if the structure makes sense in terms of sequence variation. And all of his secondary structures, which were solved by in vitro, no in vivo, just in vitro, chemical probing, and sequence comparison, these have all been validated now with X-ray crystallography. So mostly Rob Beatty's group in Boulder and Dinshaw Patel's group at Sloan have come up with 3D structures of the different riboswitches, and they're consistent, if not identical, to the predictions from Ron Baker's lab. And so our group is basically trying to almost follow in the footsteps and use a similar approach to these gigantic RNAs in humans and mammals. And so we came up with a method we're calling shotgun secondary structure determination. This is not really related to shotgun sequencing, but it has like the same feeling. We were inspired by that word, I would say, at least. And so the question is, how do we go from sequence to 2D secondary structure for RNA molecules? And the idea is, the classic problem is if you have one sequence, but two different folds or many different folds, how do you know where the right fold is? And so the normal way, like the riboswitch people would do, is to, and other groups, and also the ribosome people way back then, would do chemical probing on the RNA to get a feeling for where the base pairs are. And I have some more slides on chemical probing, but basically there are different biochemical techniques you can do where you can add different reagents to the RNA that bind only to loose, flexible nucleotides and do not bind to base pair nucleotides. So for each base, you basically get information on is it base paired or not base paired. And from that, you can try to come up with different secondary structure possibilities. And then the next step is to try to get information about covariance across species. So the idea, as Christian was talking a lot about in his talk, is that if you have mutations like going from human to mouse to rat, where a CG goes to a GC or to an AU, this is some supporting evidence that the base pair is conserved, even though the sequence is changing. You have a conserved structure. And in terms of the experiments we do in our wet lab, one of the original techniques from Ron Baker's lab for riboswitches was just inline probing. And this is basically you have your RNA and you just let it sit there overnight. And it turns out that it self-cleaves, so it cuts itself, but only in the regions that are highly flexible. So from the reaction rates, you can, the reactivity levels, you can map out which bases are base paired and not base paired. And then Kevin Weeks in 2005 came up with a technique that gives you similar information, but it's more practical. You can do it in a few hours. You can do it in a lot more different kinds of conditions. And this is the selective 2' hydroxyl acylation analyzed by primer extension or shape probing. And in this case, you add this special shape reagent, and this forms covalent bonds to the 2' hydroxyls on the backbone of the RNA, but only if the backbone is very flexible. If it's base paired, it's not very reactive. And this is another way to map out the base paired versus non-base paired information. And then some of the older techniques used for ribosomes are DMS probing. And here you add DMS, which is reactive to the faces of the bases for A and C bases. And then finally, there's another technique, hydroxy radical probing, which gives you information about tertiary structure of the molecule. And so all these techniques are really good for small RNAs, but what about for large RNAs? For large RNAs, it's a much more complex problem. So for the smaller RNAs, if it's, say, under 100 nucleotides, you can basically have a probing signal, and sort of just by looking at the signal, you can almost figure out what the structure is. For large RNAs, it's a different situation where you may have many different folds corresponding to one sequence. I think it scales as n to some high power. I forget which power. And so the idea is that you have all this chemical probing data about which if you're a base, you know if you're base paired or not base paired. So if you know you're paired, the question is, where is my partner? There may be many other possible partners. So it's not trivial to figure out where the partners are. And so an analogy to this is, suppose you're planning a wedding and you have people coming to your reception, and you have different families and different factions, and they may all not get along, and you want to make sure that at least there's no all-out wars in the reception. So you have to figure out all the permutations of who can sit next to who. And then, if you can imagine, if you scale that up to thousands of people or nucleotides, it can be really hard to manage. So the solution we came up to solve this problem is, the last note on this I wanted to mention is that in addition to this complex situation, you may also have this sneaky person going around switching name tags. My brother did this at one reception where he wanted to sit next to someone, so he switched the name tags, and then that can screw up the plan. So we'll get back to this later. So the way we solve this problem is we call this shotgun secondary structure, or 3S. And what we do is we do our chemical probing like everyone else does for the entire RNA. But then in addition to probing the full RNA, we do successive rounds of probing of fragments of RNA. And so we divide the full RNA into smaller fragments and then redo our chemical probing on those smaller fragments. And the idea is that if we can find a match for the chemical probing signal for a sequence region in the context of the full RNA that matches it in the fragment, this is really good evidence that it's an isolated subdomain. And so it's a sort of divide and conquer technique where if we can find enough isolated subdomains, we can fold those more easily and then put them together. And then in some cases, when we're lucky, there's only a few bases left over, which give us the long-range interactions. So we've done, I think, 5 or 6 of these systems, and it's worked for probably about 5 of the 6. There's one where it was really tough, and it didn't quite work, this technique, but it's been successful in certain systems. Okay, so just to give you a summary of the technique. Yeah, sure.
With respect to the last slide.
Yes.
So if you think about, if you display these RNA structures not the way you display them,
yes,
namely as planar graphs, if you display them as diagrams,
yes, right,
and you would put the sequence positions on the x-axis, and you would draw the bonds in the upper half plane.
Okay.
Right? You would get in what is called the non-crossing diagram.
Right.
So now, the folding is so fast,
yes,
because you can recursively do it. So you use the fact that once you've drawn such an arc, there's something below it.
Okay.
There's something above it.
Okay.
So it's clear if something's below it and you can't cross,
yeah.
then what is in there has to be, again, the same type that you're looking at to begin with. And if you were to collapse everything that is inside of that arc, you can just collapse it, and you would still have, again, an RNA structure.
Yeah.
So if I would entertain this for what you're doing here, is once you have stipulated the first one to be drawn,
yes,
this means you commit to an arc.
Right.
And now your problem becomes exponentially easier.
Yeah.
Because you have an inside and you have an outside. So you could actually almost mimic algorithmically your probing for non-coding RNAs. I think it's an afternoon work for Thomas to figure that one out.
Yeah, this sounds like a great idea. Yeah.
Because then you can really fast.
Yeah, and we've only looked at things between 500 to 1,000, and for going to big, this sounds like a great idea to go to large systems.
And
I think
the energy is local in the sense, it's loop energies.
Yeah, right.
Typically, which is responsible for this. So the whole recursion method should be completely compatible and localizable, which would mean that you have a polynomial time algorithm.
Okay.
I guarantee you.
All right.
You have a polynomial time algorithm sitting there.
That sounds like a great idea. Thanks. All right.
Yeah. Whether or not the nucleotide is free or not.
Yeah.
You're not getting base pairs.
we're not getting
We're not getting a base pair.
That's true, yeah.
We're getting information about this base is involved in a base pair. You don't know what it's
Who it's,
part of,
yeah,
what it's,
this is
what,
true.
Whom it's marrying.
Yeah, yeah, exactly, right.
So it's a little more complicated. And these pieces are overlapping, of course.
Yeah, we try.
Cool.
Okay. Okay, I guess I'll keep going.
Because you think on the one that you said
Oh, yeah,
that didn't work, right?
Yeah.
Do you think you fragmented this in such a way that you disrupted, thinking back to your very long-range interactions with the entire thing?
Yeah, yeah. There are
Something in the
some really pesky fragments that we think weren't cooperating, and yeah, so it was something like that. We haven't really explored, these studies take a while, at least three months, if not more, per RNA. So we've only tried a few different schemes of fragments. I have my own that I thought of that were more mathematical, and then the postdocs have some that are easier to do. So we have maybe 4 or 5 strategies we've tried. So we haven't done a systematic study on what is the best approach to doing this, I think. But yeah.
I think you mentioned it. What is shape activity exactly?
Shape is the you add this special reagent called NMIA, or in the one that works best in these situations is 1M7, and this it reacts with the 2' hydroxyl. Sorry, this one, and it reacts if the backbone is very flexible. And the reason for that no one really understands, I would say. But they've done a lot of controls against crystallography structures. Yeah.
So if it's high, then is
If it's high, it's not base paired, and if it's low, it's base paired. Yeah. Okay. Okay. So just to give you a summary of our strategy, which is largely modeled from Ron Baker's strategy. We're studying every RNA one by one. We do the chemical probing, and then to deal with the long length, we chop it up into fragments and redo the probing. And then if we have data in other species, we get in. We do some sequence alignment using synteny. So if we look in other species nearby the related genes, and then we use the structure to improve the sequence alignment, and then we try to iterate this to get a secondary structure that looks similar across species and is also consistent with the chemical probing data. Okay, so now I'll go through some different examples we've done. Whoops, this is kind of. So the first is COOLAIR. And much of this work was done by Irina Novikova, who's a staff scientist now at PNNL. She was sort of a tour de force and did tons of these long RNA probing studies and folding them and everything. And so in this case, we collaborated with Carolyn Dean's group at the John Innes Center in the UK. And Carolyn discovered a long non-coding RNA called COOLAIR. And this is an interesting RNA in that it's epigenetic, in that it's a cold-induced silencing of the polycomb target, FLC. And it's sort of a repressor of a repressor, where in the fall, there's FLC, which basically stops flowering in, for example, tulips or Arabidopsis thaliana in plants. And then in the springtime, this RNA comes along and it represses that repressor and allows flowering to happen again. But it only works if it's exposed to 4 to 6 weeks of cold. So it's like a cold-dependent functional RNA. And so Carolyn was interested in trying to figure out the secondary structure of this RNA, so she came to us and we started working together on it. And so we applied our shotgun technique to the COOLAIR system. And so this is our probing data for the intact full-length RNA. And then in this case, we did 3 fragments. And in the first fragment, we found a case where we saw a pretty good match of signal for the sequence in the context of the full RNA and then in the context of the fragment for this region. But in this region, it was kind of mismatching. And so in the but this was good data supporting that this part is probably a modular secondary structure fold. And so we could form this three-way junction from this probing data. And the way to read these diagrams is these are the RNA base pairs, and then the colored nucleotides have high shape reactivity and are likely not to be base paired. So that was that domain. And then for this fragment, we saw good agreement across almost the whole fragment, and then enabled us to come up with this form of the secondary structure. And then finally, when we can put everything together, we came up with this secondary structure for COOLAIR. And so we have the three-way junction. We have this other domain from the other fragment, two long helices connected by a big loop. And then in the middle, we have a multi-helix junction, a five-way junction. And then interestingly, in a lot of these RNAs we're looking at, we've seen this motif where we have a large loop on the 5' side, and on the other side we have almost no loop at all. So we often see in the long non-coding RNAs these motifs with between 14 to 22 nucleotides here, and then between 0 to 4 nucleotides here. And we're calling this the right-hand turn motif. And it's often full of purine bases. And so in COOLAIR, we see two of these turns like this. And so next, what we did is we found, we used this secondary structure to search other genomes. Before we worked on this, people thought COOLAIR only existed in Arabidopsis thaliana, but we found this in 5 other genomes with pretty similar secondary structures from just sort of without using chemical probing. We could see similar secondary structures in the other species, and then we went ahead and did the chemical probing on one of them, Brassica rapa, and we see pretty similar features between the two species. So at the top, we see the right-hand turn, most of the bulges are pretty similar and so forth along here. And we have a multi-way junction, and then also the right-hand turns there. So we think that this secondary structure is conserved across Arabidopsis thaliana to Brassica rapa. And then in Carolyn's lab, her student Emily Hawks did RT-PCR from different species of Brassica using primer sequences predicted from our secondary structures, and then they could find the transcripts. And then this is showing examples of this right-hand turn in different RNAs. So this is in the COOLAIR here. And then also we see this in another long non-coding RNA that I'll talk about in a few slides from now, steroid receptor RNA activator. And then we see this a few other places, but it's really rare, I'd say, in the U6 RNA for splicing. And then in the ribosome, the ribosome does have a turn that looks like this, but we're kind of ruling this out from our scheme of classification where we want it to be larger than 14 nucleotides. But there is one big loop in grasshopper mitochondrial ribosome, but it's hard to find across ribosomes and in the other RNA secondary structure databases. And so these are the secondary structures for the other species. This is Arabidopsis lyrata, C. rubella, A. alpina, and this extremophile one as well, and then Brassica rapa. And then from these, we put together a consensus secondary structure map. And in this consensus, we're only calling base pairs a base pair if it appears in all 6 species. If it's a mismatch like an AA in any of the one, we don't include it in the structure. And so from this sense, we see the conserved features across the different species. And then finally, we have a little bit in the paper on single nucleotide polymorphisms, where it turns out that for Arabidopsis thaliana, there's variation in the phenotype across Europe. So in particular, in northern Sweden, there's a specific variant that has much later flowering than the normal case. And so we went and we did the probing on that, and we saw that even though the sequence is actually quite different, we get a similar fairly similar secondary structure. So there's a large insertion in this variant, but we still get a similar secondary structure there. Okay. Okay, so next I want to talk about Braveheart. And in the Braveheart case, this was a great collaboration we had with MIT, with Laurie Boyer's group there. And Laurie discovered an RNA called Braveheart, and she showed that this is required for lineage commitment for heart cells in mice. And so in this case, it's about 540 nucleotides long. And then so she wanted us to look at the secondary structure of this molecule. And so again, we applied our technique to this system, and we were able to come up with this secondary structure where we have, again, this right-hand turn motif here. And in the paper, we call it agile, asymmetric, G-rich internal loop. But it's also the same as the right-hand turn motif. And then we also have a multi-way, 5-way junction here in this fold. And so after we got this secondary structure, Laurie, we were trying to figure out what to do. And she's like, well, which structures do you think might be interesting to look at? Well, we said, well, we see this in some of the other long non-coding RNAs. So, so she ended up doing CRISPR experiments on this system where she knocked out this right-hand turn motif with CRISPR to see what would happen in mice. Does it affect the cardiovascular development in the embryos? And then in addition, she disrupted this helix H4 to see what that would do. And so what we did is this is just highlighting the different domains, domain 1, domain 2, domain 3, and domain 4. And so we then, and this is the right-hand turn, right? This is showing this large asymmetric turn with a lot of purines in it. And then the nomenclature here is just that again, the colored circles are high reactive in shape. And then in addition to shape, we used another reagent, and these are the stars here. DMS are the stars. So after, while Laurie was doing her CRISPR deletion of the loop, we also deleted the loop and looked in vitro using chemical probing. And these were all experiments done by Scott Hennelly, and Irina did some of the early experiments in this system. And so interestingly, when we deleted the loop, we found that the structure is basically identical, but the loop is missing. So that it's not too surprising, but it's kind of surprising because when you just delete something, you kind of have no idea what it's going to do to the structure. You know, you could get a completely different fold. In this case, we just deleted the loop, and we have exactly the same secondary structure, except the loop is missing now. And so then when Laurie did her CRISPR experiments, she found some interesting results where she basically found that if you delete that loop, it takes away the entire phenotype of Braveheart. And so she has an assay for embryoid beating bodies. And then this is showing the percentage of beating bodies that are contracting, kind of like heart muscles. And then in the wild type, you have the wild type value. And when you delete this right-hand turn, it really diminishes the activity. And then from there, she and her postdoc, Zhu, did protein microRNA and RNA IP analyses, and they have data suggesting that this loop interacts with a protein called cytoplasmic nucleic acid binding protein, and it's a zinc finger. So this is interesting that there's some data suggesting that this could bind a protein, this loop. And then in the control also, when they tried to disrupt the helix 4, this had no effect on the function, but disrupting the right-hand turn loop did have a big effect. And then this is just showing that they did get high levels of Braveheart transcripts with northern blots, and also the mutant with RT-PCR, and then again for this percentage of embryoid bodies, it's way down for this piece that's missing the loop. But in the control where they modified a helix, it's more wild type value like. And then they also looked at the effect of deleting the loop with PCR with qRT-PCR for cardiomyocyte markers. And so in the end, they're concluding that this loop within Braveheart is really critical for cardiovascular lineage commitment. And this is showing Laurie's beating embryoid body assay. And so this is wild type, and then when you knock out the loop, you no longer see the beating happen. Okay, so then I'll go on to the next example, and this is this steroid receptor RNA activator. This again was work done by Irina Novikova, and this is an interesting RNA that helps co-activate the estrogen response in humans, and it also binds to many different hormone receptor proteins as well as other proteins. And in this case, this was the first RNA we looked at, and we used several different probing techniques to sort of cross-check. We did shape probing, we did inline probing, we did DMS, and we also did one called RNase V1, which is sort of the inverse, where RNase V1, instead of being reactive for the looping regions, it's reactive in the helical regions. It's an enzyme that attacks RNA helices. And then again, for the fragments, we see pretty good agreement for some of the fragments, and that allows us to get these modular secondary structures. And we came up with this secondary structure diagram that's consistent with the chemical probing data. And so we're breaking this into 4 domains here. And then other groups, structural biology groups, have looked at some of these domains. Karen Schmidt has shown that this region here binds to the HAT acetyltransferase SRC1 protein. And then a group in Switzerland has looked at, shown that this has a high binding affinity with the Sharp protein here. Yeah.
So when you interpret the chemical probing data
yes,
with
right.
you call it the shotgun method, with this overlapping thing,
right.
So do you know whether your solution is unique?
No. Yeah, there could be other solutions, I think, yeah.
Because from what you show me, everything seems to be modular, right?
Yeah.
But that is also already when you started the method, right?
Yeah.
When you do the shotgun thing, then you're automatically looking for modular things. So I'm kind of wondering whether the data, leave room for interpretation for genuinely non-modular solutions, like ones where you have a pairing from the first to the last. Say you have a huge arc.
I mean, we can have that. So, so basically, the fragments don't. Like, for example, for COOLAIR, I showed one where half of the fragment had a match and half had a mismatch. Right. So for the half that had a match, we see nice and modular, but for the other half, that leaves room for a big arc like that, I think. Yeah. Okay.
So you have any idea about the solution space that your algorithm is going to give you? You think there are hundreds there? You think they grow exponentially?
The
data and your confidence in the data?
I'm not sure. I don't know. I would say, I don't know. That's something we need to look at, I think, Okay. Okay, and then, so this is just showing some of the CAP-BP data for this region here. And then again, we see this right-hand turn motif as well in the SRA system. And then if we do, now this is an unusual RNA in that this RNA is both coding and non-coding. It has at least 5 different splicing isoforms. And we did the secondary structure of several of the different isoforms, and they're pretty much all identical with the exception of insertions of a few helices. So because it's coding and non-coding, it's really easy to do the sequence alignment, and then we can look at the conservation across the higher organisms. And this is just colored by the different structural domains. And then this is showing the same system with the different probing methods. This is shape and inline probing, and then with DMS and the RNase V1, and we see consistent. So the secondary structure is consistent with all 4 forms of probing. And then finally, we see that the looping regions tend to be highly conserved by sequence and tend to have a lot of purines in them, which is similar to a lot of other functional RNAs like ribosomal RNAs. Okay, and then the final system I'll talk about is the Gas5 system. And then in this case, this is an RNA that's important for growth arrest, and it's Gas5 is growth arrest specific transcript 5. So it's important in T cells, and it's a repressor of the glucocorticoid receptor. And then this is a collaboration with Andrea Allen's group at University of New Mexico. And in this case, Scott Henley came up with a way to port this onto the Illumina platform. And so these are our results, just comparing the CAP-BP versus Illumina. And we saw a pretty good agreement between the two methods for both shape probing on the top and then for DMS probing on the bottom. And then we see, in this case, we did not see these different subdomains here. This was more just a string of different helices here. Yeah. Yeah. Uh-huh.
So, I don't know if I just ask this in the most naive
Sure.
way. Is there a way to quantify the agreement other than just to look at these things? And what do you?
Yeah.
How do you do this?
So we calculate a correlation
You do it through just a
function.
straightforward-
Yeah, just through correlation so far. That's what we've done.
Thank you.
Okay, and right, and then this region here, which has been shown to interact with the hormone receptor, we see to have structure in that region. And so the next step would be to do binding assays to see if the protein actually binds in an in vitro sense here. Right. Okay, and then so finally, I just wanted to say a few words about there are many groups doing in vivo structural probing, and this is a question we often get, how come we're not doing in vivo probing, and we're moving to that. But one sort of cautionary tale is that in vivo, the system is much more complicated in that you have many proteins binding to the RNA, most likely. And so this is just a schematic showing that if you had a reactivity profile that looked like this, where you had a red looping region and another red looping region, one possible fold could be this, a three-way junction with two looping regions. But if you have a protein in the system, another solution could be a protein bound masking that region and then two looping regions here. Another solution could be a protein bound here and then two flexible regions here. Another solution could be a protein bound here and two flexible regions here. And so then if you scale out to, say, 10,000 nucleotides and you have hundreds of proteins, it's going to be very, very difficult to come back to a secondary structure in that environment. So we're advocating for people to do an in vitro system first, where you only have RNA present, and then try to get a framework for it, and then go for the in vivo system. And this is kind of analogous to that person in the wedding reception switching the name tags, where you have something that's kind of making your life a lot harder, basically.
All those
Yeah.
different shapes wouldn't have the same folded free energy stuff. I mean, maybe they're all possible, but they don't
Yeah.
have the same likelihood.
Well, I think we could argue about how good the free energies are when you calculate those free energies, I think, I'd say. Yeah. Okay. So just to wrap up, we're seeing that the fragment method helps find these modular subdomains. And then by process of elimination, it makes finding the long-range interactions easier. And that in the case of the COOLAIR system, we saw similar secondary structure features in two distant species. And then in our collaboration with Laurie Boyer's group, her CRISPR experiments show that this looping RNA motif, the right-hand turn, is essential for RNA function. And then finally, we believe the in vitro probing shows a framework for in vivo studies. And then I just have two more slides, because I wanted to highlight some of our new MD work we have. I'll just go through this really quick. We've done the first simulation of an entire gene locus. This will be the first billion atom simulation in MD. And this is work done by many, many people in our group, Tamar Schlick at NYU and Yuji Sugitas at RIKEN in Japan, and Wataru Nishima's in my group, and he's doing much of the heavy lifting on this project. And we looked at the GATA5 gene. This is based on a coarse grain model from Tamar Schlick's group. The run required 500,000 cores, and we're using a new code from Genesis that allowed us to do this run. And then here's some of our early results of some of the early structures coming from this study. Okay. So then I'd just like to thank Irina Novikova and Scott Henley and Wataru, who've done most of the work, our collaborators, Carolyn Dean's group, Lori Boyer's group also, Cheng-Shung Tung, and Christian Spahn and Paul Whitford, and the funding agencies. And finally, we're looking for 2 postdocs now, and you for your attention. Thanks. Yeah.
This may be out there, but thinking about the last talk and the distillation of the data down into feature sets that then could be fed into a machine learning schema. Do you think you could, does your data set in terms of the structure that you determine empirically with the different methods lend itself to that, or is it rich enough to really predict a structure within
I think it's a start, I think for sure, and data sets like these would be great to put into machine learning algorithms, I think, yeah. I'm not sure, I haven't done a lot of that myself, so I'm not sure how many data sets you need to get to have success with that, I guess. But.
So these
Yeah.
segments that you have overlaps, so the function, can the function be different at the overlap between the 2 segments?
I didn't quite get it.
This function that
Yeah.
tells you whether it's likely to have a base pair or not. Okay?
Oh, yeah, right,
Each
right.
segment. And then
Yeah.
the segments have overlap, so can the 2 functions be different at an overlap?
Which function? I'm not sure. I don't get the function.
That shape.
Oh, okay, yeah. If you're using 2 different probing methods,
Not the same problem.
the same probing method, then
Can these 2 functions be different at the overlap? Let's see segment.
I'm not sure.
do you mean the biological function or?
It's a biological function.
Oh, no, sorry, yeah.
What do you mean?
What do you mean? Do you mean a mathematical function or the biological function?
No, that graph, that shape.
The reactivity or reactivity?
Yeah, exactly.
Oh, so
So what I'm saying is that can this reactivity be different for the 2 segments at the overlap?
yeah.
What does it mean?
If it's different at the overlap. So these, for example, the one case where the fragment method did not work very well, each fragment is going to have some kind of fold. And so, well, even for the example of COOLAIR, there was the one example where part of it matched and part of it didn't match. So in the part where it didn't match, in the fragment, it makes some other fold. So it just means that it folds the fragment. That portion of the fragment has a different fold than in the context of the whole RNA. And likely, if you have 2 fragments that overlap, and the overlap pieces have 2 different folds, it just means that that portion of the fragment folded differently. So for example, you could have a fragment with a 3-way junction that's the same in the fragment as in the whole system, but then in addition to the 3-way junction, you could have like 2 other helices that are folded in the fragment, but they might be unfolded in the real system or something like that. Does that answer your question or not really? Okay, we can talk offline, I guess.
Hi. So actually, I think the original thought when super long sequence is because there are like a combinatorial explosion.
Yeah, that's right.
The possible structures.
Yeah.
But then instead of like doing things like subsequence by subsequence, it seems to me that basically we can look up like all possible cases and do kind of like inside-outside thing. So instead of like, so you can use the global signatures who is paired, who is unpaired, and then figure out, okay, what are the subsequences that is best to check to knock out the other possibilities and do this recursively instead of just looking at
Yeah, I see. Yeah, I see. So more like even more hierarchical. You're saying like that. Okay.
Like, of course, we have this like. I think this is some of the things that our group is really good at. Like, we can develop this kind of like strategy to look at what is the next sub.
Yeah. Right. Yeah, I think that makes sense. I think so. Yeah.
Technical question to follow up on what you were asking. When you fragment it,
Yes,
non-specifically, so
right.
you're almost selecting for a intact domain into your fragment.
Yeah, but we compare it to the data from the whole RNA. Yeah. Right. Yeah. So yeah, because since we compare it to the full RNA, it basically shows that there's a modular domain in the full RNA. Yeah.
You're selecting almost for modular domains.
Right. Kind of, I guess.
Kind of,
Yeah.
because of the way you fragment it.
Oh, I see what you're saying. Yeah, I mean, yeah, I'm not sure that's the case because when we do the probing on the full RNA, there's no bias in that at all. You know, we just probe the whole RNA. There's no bias for domains anywhere on that.
No, but
Right.
there's no bias for them. Yeah, but I mean that's a shape. The chemical derivatization, well, it's biased for loops, but when you fragment it, it's in extended regions, right? That's where it clips, right?
No, we can clip anywhere we want. Yeah. So we're just synthesizing our RNA like from runoff transcription.
What do you fragment with? What is the?
We're just like we're just doing runoff transcription. So we're just making different RNAs. Yeah, like that, and then folding them. So.
More questions? That's not the case.
Okay.